Hello, welcome. Thanks for being here this afternoon. We are doing a deeper dive into biology. I am Krista Sampson, your moderator for this webinar, and I am here with Victor Sampson, the presenter and author of the biology book. And so he's going to show you a little bit how to use the team system, and then he'll dive in. So I will hand it over to him.
Today we're talking about high school biology. We are really excited about our high school materials. For biology, we have two books. One is an NGSS version that has 8 different investigations that is everything from cell function to systems to evolution. And then we also have one for Texas. Since the standards for NGSS and the TEKS don't quite overlap, we wrote 2 books. So we have them perfectly aligned with both sets of standards. So in the Texas version, you have different investigations than you do in the NGSS version. Today, I thought we would dive into investigation we call tissue repair. I know I started my career as a biology teacher, and one of the units you're always on, or one of the topics you're always dealing with, is mitosis or cell division, and how you can go about teaching that. So I want to show you a version of that you can use from one of our books here. So let's go ahead and get started. So as in all of our investigations, we introduce a phenomenon at the beginning. So instead of starting off with, here's the definition of cell division or why cell division is important, we couch this into something, we frame this around something that kids can see in the world around them. So we introduce that phenomenon, we do it through a video. I'll show you what that looks like here in a moment. Then we ask kids to talk about things they noticed during that video. As they then share out those things they notice in small groups into the class, you as the teacher kind of wait for them to start talking about something related to the task of the investigation. And then you can move into that section and introduce this idea that how bodies repair themselves from damaged tissue, and put in the wrinkle in if how do we have a copy of all of our genetic information in our new cells that we have to create to be able to repair tissue. So then we introduce that guiding question. From there, we ask each individual student to take a moment and draw a model or create a model that they can use to try to explain how your body makes new cells from existing cells and how does that end up working. So let them start drawing that out and thinking about that. You have them share their models within their groups, and all of a sudden a bunch of new questions starts coming up in each of those groups as they're trying to figure out the things they not sure about or disagree on. Then you let each group come up with some new questions that they have now based on what they don't know about this phenomenon. So it's a great way to get them hooked into the investigation. All of our books come with a book code that allows you, once you have an account in the Learning Hub, which is absolutely free, teacher account, you create that with your name and email. You enter your book code that comes with your book, and then that gives you all the investigations in the book are put into your library and Learning Hub. And then what you can do is a couple things. One, you can download all these handouts in color from those investigations, but you can also take advantage of something we created called the presentation mode, which allows you to use Learning Hub as a slide deck to facilitate. So you're able to move students through an investigation by just plugging in your computer to a smartboard or projector, and then opening up this presentation mode, and then you can just kind of move through it. So for example, on this shows a screenshot of what that looks like. And you'll have here the same text that's on their handout. So you can kind of read this aloud or have them following along. But most importantly, it also includes any of the videos, like the phenomenon that we've got here, so you don't have to try to dig that up. Let me show you what the phenomenon video looks like for tissue repair. And you can actually show that a couple times to them, and they point out things that they notice about from where you end up having a pretty good cut to scabbing to new tissue and a healed body. So you start thinking about, well, what happens? How's that go with that? As I mentioned earlier as well, in the Learning Hub, you also have parts that match the handout. So then you can say, hey, share what you noticed on the video with your team. This is where you should be writing this on your handout. So it's a great way to keep everybody on the same page and not getting lost during one of these investigations. From the task stage, now we jump into the ideas stage. And what we're trying to do now is introduce some science ideas, disciplinary core ideas and cross-cutting concepts into the mix that students can use to help answer the guiding question. So these usually are that help them answer their new questions. So they don't answer the question of the entire investigation, but the questions they have about their model that they're trying to figure out. So in this case, we first introduced the idea of cell structures, right? So things like membrane, cytoplasm, nucleus. We also talk about genetic information inside cells and how that's organized. Talking about different chromosomes that we have, so they can kind of see these different things that make up cells. What after each of these texts, there's always a little box on their handout that asks them not kind of analysis questions, but for them to read over the text to decide as what's going to be useful or helpful for them figuring things out. And we do this intentionally so kids can start getting used to this idea of that. I'm just not reading to find an answer to a question. I am reading because I need information that's going to be useful, and not everything is always going to be useful for what I'm figuring out at this time. What I'll often do is when I'm first working with a group of students, especially the first time they're doing this, I'll usually read it aloud, have them follow along, and then have them in small groups identify some things that they thought were really useful. Those then are shared out to the rest of the class. Every group shares out a few of those. We can make a class list on the board, and then students can add anything that they missed before. So it's a nice way to make sure all those ideas are there. The second idea for this, we use images of cells creating new cells. So these are images of cells in the process of mitosis. We don't introduce that name specifically yet, just so they can kind of start thinking about this as a cell division and there's different things happening, and we're not sure what order they happen in, but different things are happening here. And then we finally introduce the idea of using models to help represent processes, right? So that you can use a physical model or a simulation or any number of different things that allow you to test ideas and try to make sense and match to pieces of processes or incomplete processes that you can't fully see when you're investigating something small, say like cells. What we do in the Learning Hub too, what is once again, you have all this text so you can read it off the screen instead of reading it off the handout. We also include all the images that are on the handouts with an added feature that you can click on them and blow them up to full screen. That's nice for two reasons. One, often when you're using handouts, you end up with black and white copies and you can't see all the details on it. And then two, it's much easier to magnify these pictures, and then you can point out things that are happening in the image that you want to bring up as you're reading this aloud, or the students are ended up reading this aloud. So that's a screenshot from Learning Hub there that allows you to do that. Once again, you also have the bubbles, we call them the bubbles, that students will add in there. So while they're talking over what's important, you can put that up on the screen and say, keep everybody on the same track or same place. After they have these ideas, then you move on to the plan stage. You start off the plan stage by introducing the materials they will have to use during this investigation. In this case, they're going to be using pop beads with magnetic connectors to represent chromosomes as a physical model, and then they'll have cards that they can use to see the different stages of mitosis or the different stages of cell division. They're intentionally not ordered in the correct order. We just call them events A, B, C, or D. Part of what they'll have to figure out is what order these go into, and then use the beads, the chromosome models, to try to figure out what's happening inside these cells in these images. We also include that in the Learning Hub so you can see those right there. I always like to have it on the screen behind me as well as on a table so I can kind of hold them up and show them what they end up looking like, so everybody's on the same page. Once they see what materials they'll have to use, they can go ahead and create a plan as a team. What do you plan on? And in this case, it's what's your question you're trying to answer, what observations or measurements do you need to make sure you keep track of, how are you going to analyze those once you have them. So it's not just play with the materials, but what kinds of information you need to seek out. I have groups of students do these as teams, and then they can share out with me if I'm okay with their plan, sign off. If not, I can give them feedback. So it's a nice little check-in right here to make sure everybody is on not on the same page, but everybody's going to have a productive investigation. And then you can give them a little nudge if you think they need it. After they have their plan approved, they have the materials, they know what they're doing with them, so they go ahead and start creating, trying to figure out that model. On their handouts, then they have a place to record data or observations or ideas that they're having as they're going through. Once they've collected the stuff and worked out their model with the physical equipment, they can then do more of a conceptual or visual model that they can keep track of. This is how they're going to be answering the question of how do cells end up dividing. Once every group has their idea, their claim, their model, they're going to develop an argument to share those out, because every team will have slightly different ideas, which is great at this point. It looks just like science. So what we do is have them make these arguments on whiteboards so they're easy to present. So in this case, they'll have the question added on there. Their claim will be a model of what's going on during cell division. Then their evidence for that, so they'll match it to like what they see in the cards and those kinds of pieces. And then they'll use those ideas from that were introduced earlier in the idea stage as part of their justification of their evidence. Once they make these boards, they can share them out with each other and give each other feedback. There's lots of ways to do that. There's gallery walks, there's modified gallery walks, there's sticky note feedback, any number of different ways. We also use we have space on the handouts where kids can write down ideas they have, either that if they've seen in other groups, they can bring back to their own, or if they've gotten feedback from another group, they can write those down as a way to kind of think through of, ooh, this might be useful for improving what we're doing. We also, in the Learning Hub, add extra kind of supports for students as they need them. If they're getting stuck on what's on the handouts not enough, you can add an extra graphic up on the screen and remind them kind of what things, as kind of some hints that they can put in there. All just little things to make things work a little smoother. After they've all shared and revised their argument, and the key is they've revised it, right? So they've taken that feedback and finalized what they think's going on. It's really important to bring everybody together into a whole group discussion. We call this the reflect stage. And you want to talk about two things. You want to talk about the core ideas that they were using, and then they also want to talk about how you engage in practices. The easiest way to do this is we include these images in the Learning Hub for you, so you can show these up. What I do is just put this screen up for students and ask them to take a look at the image. That image is actually the same one that was in the idea stage earlier. And then start talking about things about how do we know about cellular reproduction to explain what's happening in this images. So what are the things that we're actually seeing? So they start talking about things like cell structure, what are nuclei, what are chromosomes, what are cell membranes, right? And start thinking about how these things, usually what they'll do is start talking about the stages of mitosis that they were uncovered in that. So it's a great way at this point to kind of correct any misconceptions or clarify any misunderstandings. Once you're happy with their ideas about the core ideas, then you can move on to the practice discussions. At the high school level, we're really getting them to start thinking about how do you ensure that you have high quality evidence, right? So everything really in science is based on the idea of does it fit with evidence? So you've got to be sure that your evidence is of high quality. So we start having them thinking about that. So things that you did as you carried out your investigation to increase the confidence in your evidence, right? So what are the things that made you really feel good about that your evidence is valid, right? Then what could you have done differently to improve that? So if you start questioning the quality of your evidence, what could you have done differently? And then what can we as a class moving forward the next time we do this to make sure that we're carrying out the best investigations possible to generate the highest quality evidence that we can. And then they start coming up with those rules on that. In their handouts, we include a sheet, a reflect sheet that students are not necessarily taking notes on, but taking a minute to reflect on everything they talked about and writing ideas that they want to remember for their future selves. So that can either be about the core ideas that were there, things that they want to remember for the future, or about practices. So these often students start writing down the class rules down here so they can refer to them again later. So it's a nice way for kids to really be more metacognitive about what they've learned and what they need to keep in mind as they're moving forward in their science education. Finally, we have students, each individual student write a report on their own. We have them write a report on their own for a couple reasons. First is they've been working in groups or as in whole class formats up to this point. So right now we want them to have a chance to kind of do the mental work themselves. By writing, they not only learn how to communicate more effectively in a scientific way to others, but then they also are forced to think about the content, everything that they've been talking about, seeing, doing, discussing up to this point more deeply, right? So as you have to write to explain and persuade, you are forced to think about the content deeply. So this is really an individual learning mechanism is why we add this in here. The reports don't need to be very long. We just need them to think about content and clear communication. So we have them have 3 parts: an introduction, a method, and the argument. Introduction is just what we figured out. Method, how'd you go about doing that? An argument is share what you figured out and how and why you think that is the most valid or acceptable answer that you could be making to the guiding question. Once everybody's got a report, you can then do a whole class peer review. And the way we do this is you divide students into groups of 3. I always recommend 3 because this works the best for this. So say you have a class of 30 students, you would have 10 groups of 3 students. Each group then gets a report written by somebody outside their group, right? So they get 1 copy of that and one of these peer review guides. You ask 1 person at the group to be the recorder for the group. They, as a team, read the report and then go through these questions on the peer review guide and as a team answer them. Anything they answer yes to, it means that's perfect. You don't need to change anything. If they say somewhat or no, they need to give feedback to the critique that they're making on the peer review guide. We give them I always give them 15 minutes to do that first review. Then I collect the first reports. I give each group a new report written by somebody outside their group and a new peer review guide. I have them switch recorders. So a different student in that group is now recording and filling out the peer review guide. Give them 15 minutes to do that together. Collect that last report and then give them a third report, a third peer review guide, and have a third recorder. So what ends up happening with the numbers of all this, in 45 minutes, you end up having every group review 3 different reports. You've had everybody in the group be a recorder, and everybody who wrote a report has been reviewed by another group. So it's a great way to give feedback, and also the process of reviewing actually teaches them a ton because those discussions they're having about what counts as quality is really where the learning takes place when it comes to improving writing skills. That then goes back to all the students or the original authors, then they can rewrite. We include these sentence starters in the book just as a starting point because a blank page is kind of scary to a lot of kids who are not used to writing. You don't have to use those. You can have them type out anything they want to. You can have them handwrite. You can use the sentence stone. If you are going to use sentence starters, I wouldn't do it more than twice because they need to get used to writing without all the scaffolds. But we add them in there in case whichever one you want to do first kind of thing on the investigations. After those final reports are done, I ask students always to give me your first draft, your final draft, and the peer review guide that they had filled out by their peers. And then I use that, then you can use that as a rubric. you have the exact same things that there are questions, you answer those, but this time you're given a score, and you can then use that as a grade. And you've got a really nice, easy summative assessment of this learning experience for them. All right, that was a lot on this investigation. So always remember in the books, we added very, very detailed lesson plans, doing our best to format them that they're not overwhelming. Because I don't know about you, if I get like a phone book of directions, I'm not reading any of that. So we try to break this up to make it a little bit easier to kind of follow along. So they all start with an overview of the investigation, and then how do you break this thing over multiple days, right? So any one of these investigations you can't do in 50 minutes, but most of our high school periods are either 50 or 80, depending on if we have block. So we assume 50 minutes and then talk about how you break the stages up over that. And that's kind of your guidepost of what you need to get done that day. We also include all the standards, but not just the science. We also talk about language development and also English language arts, because that is so important for their development. So you can see all the things that you're helping students learn how to do with one of these investigations. We also include information about the core ideas and the cross-cutting concepts that are a focus of the investigation. This is really the kinds of things that you need to know for that reflective discussion. Do a quick read of that right before you do the reflective discussion, and you're in great shape. We also include materials, so a list of the materials that you need by class and by group. Then we also talk about how you prepare them. So sometimes we'll include a picture or just directions. Then advice for what you do during the investigation, some extra tips if those come up. We also include kits for these, so if you don't want to source this stuff yourself, you've got tissue repair as an individual one, or we've got a bundle of all of the investigations that have on as a whole thing that can get together. So you got two ways of getting through that. And then finally, is the directions for each activity within a stage. So you've got 7 stages of any ADI investigation. Within those, you also have activities. So what we break them up as by activities. It tells you about how long, what you need to do, what to give out, what to show, management tips, those are our teaching tips, and then a section called attending to equity, which is how can you better support students that need more support, like some students that may be struggling with reading, or maybe they're learning a new language, or they don't feel very included in class. So the kids that are very quiet kind of thing. So how do you bring them out and get their voice done? So that's all in there for you. So with that, we got a few minutes left. Is there any questions that I can answer for anybody at this point?
So Samantha asked, what's the best way to help students understand the difference between evidence and justification?
Oh, that's nice. Okay, so the way I usually think of it is, well, the big thing you got to think about too is what's the difference between evidence and data as well. So as a first step, I always think of be clear as data, things as you collect, right? So these are observations or measurements. Now, since data and evidence are not the same, you have to then think about how do you transform data into evidence. And you do that by analyzing, making a graph. And then you have to also interpret that analysis for other people. So with words, explain what you're seeing, the trend, the difference. That's your evidence. So the justification then becomes, why did you decide to give that evidence or use that evidence? And that's always tied to the important core ideas. So you can think about justification as ideas that you had before going into the investigation, and evidence is things that you discovered during this investigation.
Awesome. And I was just chatting with Helen, and she was asking about the Texas book versus the NGSS book for biology, both of those, and are there different investigations? So
yes, they are. I'll put them up here real quick as we're moving through this.
And also, just to add on to that, if you want to check them out, you can also go online to the store, and in the description, there should be a link so you can check that out.
Yep. So here in the NGSS, there's a big focus on cell division and genetics and evolution. For the Texas one, we also have types of cells, diagnosis of cancer, photosynthesis, mutations, and then a bunch of genetics and evolution as well.
Perfect. And um, looks like last question we have from Caleb. What do you do when students get stuck during the planning stage and don't really know how to start?
Oh, I love that. So when they're working in this groups, the first thing that helps with not getting them unstuck is having that tool talk at the beginning. So as I was showing you, show them the materials and asking questions, what might I use this for? That takes care of a lot of the problems. The next part is when they're working in groups, use a routine I call two questions and a goal. So go up to a group, ask the first question, what are you all working on? Tell me what you're thinking about. And someone will answer that. Then you say, anything else you want to add? Second question. Then based on that, give them a nudge where you think they should go. And then the goal is, hey, I'm going to come back in 10 minutes. I want to see this part done from there. Between those two things, you'll have 90% of your problems with getting kids unstuck during the plan stage taken care of.
Perfect. And I will let everyone know about the next webinars. And
Yeah, we're starting chemistry in 1 minute, so we're going to jump over to that other one. And tomorrow we're going to do physics at 4 and Texas IPC at 4:30. So hopefully you can all join us.
all right, thanks for being here. Have a great afternoon.